And today with me, we have Alexis, which is the speaker today. So we are both we are both biologists here at Bioturing. And today we will explore the fibroblast-driven cardiac fibrosis and also apply for future therapies. Okay, so I think we can start by have a little bit recap about the last week episode. So for the last week episode, we explore epithelial cell subtype population in idiopathic pulmonary fibrosis or IPF, which reveals some of the cancer risk characteristics. And as you can see in here, so these are some like highlights during the findings last week. So basically, during the episode last week, we found out that fibroblasts play a pro-fibrotic role in pulmonary fibrosis, while like epithelial cells act as like mediators of the disease. And then, oh, I'm sorry, can you go back one slide, please?
Mhm. Sorry, let me just, here
Yeah,
we go.
sure. Yeah, okay, thank you. Okay, yes, thank you. And then we found out that like pulmonary fibrosis lungs have a lower proportion of alveolar type or AT, as you can see in the like the cell composition chart right here. And then when we explore further into alveolar types, we saw that like AT1 cells exhibit upregulation in lung cancer related pathways. We also see, you can see in the AT cell to cell communication here that like the AT cells, AT1, communicate with other epithelial cells via CCAM pathway. And then like by using serotam, we found like targeting the FOSB positive AT1 cells may present like a potential therapeutic target. And that's for the last week. And now we come to the topic of this week. Okay, so this is the agenda. So for today, we will explore cardiomyopathy. And first, Alexis will give you guys like a quick overview of cardiomyopathy and fibrosis in cardiomyopathy. And then she will show you like the three main findings that we have, which we use the talk to data beta version to explore. So first, so this is kind of like a basic flow where we go first from the identify key fibroblast markers in cardiomyopathic heart, and then we investigate the expression profiles of activated fibroblasts in the disease, and then last but not least, we identify the enriched pathways in activated fibroblasts based on the enriched genes. Okay, so Alexis, I think now you can go on and like start the presentation.
Yeah, of course. Thank you, Hazel. Okay, so just before I start explaining a bit about the disease case study we'll use today, I do want to sort of cover what talk to data beta is and how it's utilizing our human database version 5. So this is how many data sets we can actually use when we're querying specific marker genes or signature genes in the entire system, how many cells we're able to really identify, and sort of more information on cell type targeting. So this is just a recap of sort of our human database and our version 5, which is the most recent one in the system. And for anyone who's joining that is actually new to the Bioturing ecosystem and hasn't used the system before, when you want to access the sort of talk to data public repository where we curate all of the public multi-omic data, this is where you would be accessing it. So within the ecosystem, you would go directly to talk to data. And the beta version, which is the one we'll cover today, and all of the new features within that beta version is sort of available here when you click on the Go Now icon. So this is the former Talk to Data version 4. So we haven't yet released it fully on this version, but you can access the beta sort of testing here in this sort of icon. So that's just for anyone who's not really used to using the system. So now we'll go into sort of why this topic is important and why we really decided to cover a session on cardiomyopathy. So this particular disease includes two different cell types, so both the dilated and the hypertrophic form. And this sort of progressive heart disease frequently leads to heart failure. And it poses a very significant global health challenge when it comes to identifying targeted therapy that would really help two forms of this disease. So this is sort of why we decided to really utilize the talk to data beta system to analyze sort of specific signature genes in cardiomyopathy. This disease is also marked by structural change, which is sort of shown within these different images of the heart, where we show in dilated, for example, you see this enlarged left ventricle, a thinning sort of the heart muscle, whereas the hypertrophic has different structural changes as well when it comes to the disease. So really our aim is to identify if there's particular markers that drive these structural changes. Can we see that across different studies in the talk to data system and really understand the complex sort of molecular alterations at the cellular level? So that's the reason why we'll also use single cell RNA-seq data for this particular case study. And some of the key critical pathological features as well that we see in this disease is sort of increased fibrosis. We see that this results in a stiffening of the heart tissue, which we'll also cover based on some targeted genes that we'll try to analyze. And within cardiomyopathy, we also want to understand how different cell types play a role in this disease. So particularly in this sort of different forms of the disease, we see that fibroblasts are a very key driver of fibrosis. And normally under normal sort of healthy conditions, fibroblasts tend to stay sort of quiescent. Whereas when there's cardiac stress or there's some sort of trigger in the heart, we see that these fibroblasts sort of turn into these activated sort of subtype form, which proliferate, and these produce excessive collagen and other sort of extensive extracellular matrix sort of issues. So this is, again, some of the key aspects that we'll try to really analyze, and how can we do this using single cell RNA-seq data. So if we go into some of the sort of recent studies that can be found as well in the system, and also will sort of show some background into what we'll try to analyze. So these particular studies showed that fibroblasts exhibited upregulation, and there's some key fibrosis related genes that were sort of shown in these studies. So some of them is POSTN, NOX4, THBS4, and collagen 1A1. So we'll really try to look at these particular signature genes, how they drive the ECM, so the extracellular matrix, and how this worsens cardiac dysfunction in different patients. And we'll try to also see how these particular fibroblasts or activated fibroblasts can also highlight potential therapeutic targets, or how these can drive fibrosis-driven heart disease. So that's sort of the focus that we'll do today. And now we'll go into sort of the live demo of the session. So since I mentioned the main focus will be talk to data beta, this particular system really provides a valuable tool when it comes to exploring particular gene signature markers across different disease stages, different patient samples. And it really allows for a data-driven approach when it comes to identifying validated targets that can be used for this real-world case of cardiomyopathy. So that's sort of why we decided to do this. So the first objective that Hazel mentioned at the start of the session was that how can we identify key fibroblast markers in cardiomyopathic sort of heart tissue, and also different areas in the tissue. So as you know, most of sort of these marker genes have been shown to be important in the left ventricle area of the heart. So we'll try to focus, how can we sort of validate this across not only one study, but several studies in the database that have different sort of patient trajectory and heterogeneity. So that's the first question. So in order to do that, again, we'll access the talk to data system through this module, which you can explore. Once you have the new talk to data beta interface, this is a new interface that you'll see when you go into talk to data beta. You'll see that it's different from the original version, or the, I guess, the previous version. So some of the things to keep in mind when you're querying specific signature genes or target genes of interest is that there's different configurations that you need to sort of take into account in the system. So some of these are sort of the ontology configuration. So that's what type of filters you're actually going to select from the database, whether that's cell type, anatomy, conditions, different sampling sites in the tissue, or also different data relating to treatment response. So these are some filters or configurations that you can add. Now, when it comes to expression values, or how these markers are enriched, or how these are scored across the different filters and the studies that are sort of the cells that are expressing these genes, there are two scales that you'll need to sort of decide which ones you're choosing. One, which is the absolute scale. So this one within the ontology map means that when you're selecting particular genes at the absolute scale, you'll have the average rank and percentage from 0 to the largest values, but this is based on sort of the 5,000 rank that we have, so our normalized unit within the system. If you sort of want to look at not only sort of a broader insight of these expression patterns, but you really want to look at a relative mode. So you want to look at how these markers are specific to fibroblast specific conditions, then we would recommend to then change that configuration to relative. So that instead of doing sort of a normalized ranking from a 5,000 rank to 100 percentage, what you're using is that the nodes will be scaled from minimum to maximum values within that filter sort of configuration that you've added in talk to data beta. So this is the accumulation and the scales that you'll need to sort of keep in mind, as well as what sort of intensity or what AUC score you really want to identify within all of these studies in the system. So the more sort of increased AUC score that you sort of filter, the more accurate or very specific sort of nods you'll sort of have as a result. So you'll really see where these particular genes are expressed within what cell types and within what studies specifically are showing this expression profile. So these are some analysis you can sort of gather just from querying specific markers. You can look at what studies those results are coming from the system. When you click on those studies, it'll navigate you onto what sort of data sets you can actually explore further, especially if you want to do more advanced analysis or sort of exploration of the studies. What else you can do is, in this case, since our main question is how can we identify fibroblast markers in different areas of cardiomyopathic sort of heart tissue. So in this case, what we want to do is specifically select the conditions that we're particularly interested in for this case study. So in this case study, we want to look at samples relating to dilated cardiomyopathy and also hypertrophic cardiomyopathy. So we'll add these sort of filters in the system so that we're able to really look at how there's differentiation within these sort of targets. So here's our hypertrophic. We'll also want to add normal. So some of the questions that you can also answer is how can these marker genes be targeted, and can we see differences from normal sort of healthy control samples within the heart sort of tissue region that you're focusing on. So once we have our condition selected, we can then go back, for example, to what sort of cell types we're focusing on, or if we're focusing on a specific heart tissue area in the sort of studies. So when we're focusing on anatomy, this is where you can add heart tissue as a whole. So you can see the expression across the entire sort of heart sample. Or you can also look, for example, if there's differences in right ventricle versus left ventricle heart sort of regions. So once the anatomy filter is sort of loaded, you can add what area of the anatomy or within that sample are you focusing on specifically. Okay. So in this case, we want to add the left ventricle to see if there's any specific enrichment in that area. So here's where I'll add that specific filtering. And then the last step is what cell types are we actually focusing on? So since we want to look at fibroblast-driven target expression, I'll make sure that all of the cells across these studies relating to these samples are specifically fibroblast cells. So again, you can add multiple cells. You don't have to only focus on a certain group. But this is just one example of how you can query and select your filtering sort of method. So from here, we have now our filters, and we can look at other information. So every time that you select a standardized ontology label, it'll automatically generate that result. So without having to select the cell types, I can already see that these particular markers are very specific to fibroblast cells in the cardiac tissue. And I haven't selected any specific sort of fibroblast subtype, so I'll just leave it as fibroblasts. If I want to see how this relates to treatment response or other ontologies, you can also generate that result real time in talk to data beta. So I think previously you would have had to use talk to data, create a custom data set, or use other information and wait for that atlas to be generated. But now we can just generate it real time from any standardized ontology. So it seems there's no fibroblast cells where there's studies specifically having treatment response data. But again, you can select any metadata to generate that result if there is metadata for those studies. So now that we have our filtered categories and we want to actually visualize this in different ways, we can use other icons or other functions in Talk to Data Beta. For example, the composition, which gives you sort of information on sort of percentages and also proportions within these different groups and how many cells within these sort of nodes are actually expressing these genes. So if we go to the composition, every time that you generate the ontology filter, you'll need to perform a new analysis so that it automatically creates that visualization for you. And again, you'll need to also make sure how you're sort of generating those metadata labels, what's the order of the labels that you want to actually see. So you can look at within the left ventricle area, how do these fibroblasts express within these different conditions? So that's sort of a sort of question or a different sort of overview that you can generate. And again, this might take a few seconds to load within the entire system, but I can open up another example that I already have, just so you're able to visualize that result a bit quicker. Okay. Okay, so I'll go back. I'll just look at a particular gene here again. I'll search for that gene, and that opens up our Talk to Data Beta. From there, we'll go again to the composition feature. And again, we can look at analysis that we're particularly filtering within the region, or I can look at if this is done as well. So for example, here, what I want to do is actually select the anatomy. So that's the heart, the left ventricle area. I want to look at how these fibroblasts are related or the expression is differentiated across different conditions. So I can apply that search so that it sort of looks at it easier and you can see within a specific category how these genes are expressed differently. From there, again, you'll need to make sure what scale you're using. So the absolute is more of a broader insight. It doesn't particularly look at the expression within the minimum and maximum values within these filters. So if you're looking relative to what you're actually inputting, you need to change that scale so that it particularly shows that expression of those targets. And again, the system might take a few seconds to load those results, but it shouldn't take as long to sort of change that configuration. So here what we can see is that NOX4 that was mentioned in these other public data sets that were recently sort of published looking at these same questions, we can see that particularly in dilated cardiomyopathy, you have this higher AUC sort of expression within this NOX4 gene. So this particular gene is very specific to dilated cardiomyopathy samples. We can also see that there's also some level of expression in hypertrophic cardiomyopathy, but definitely there's very specific sort of reference to dilated forms of that disease. Now, if we look at other markers that were also quite important, for example, collagen-related markers, we can also see that there is also expression in dilated forms of that disease as compared to normal healthy control samples. So we don't really see these POSTN, which are very specific to driving fibrosis within fibroblast cells. And we also don't really see a high percentage of cells that are expressing these collagen-related markers. So we see them rather in these other forms of cardiomyopathy, which does beg the question that normally in these conditions, you have this increase of collagen in particular areas of the heart tissue. So these are sort of insights that you can gather just by looking at Talk to Data Beta and filtering specific sort of ontologies. Okay. So the next question from here is how can we then move into the next objective, which is how can we investigate expression profiles of these activated forms or these activated fibroblasts, and particularly within these diseases or this context that we've sort of covered today. So for this sort of question, we'll normally need to use differential gene expression to really identify what particular targets are differentially expressed at a particular significant score level. So we'll have both the two different spectrums, which is what markers are upregulated within these activated fibroblasts and which ones are downregulated as well within these different tissues. So for that, I'll go back into our sort of analysis, our ontology, so that we're able to look at that particular question. So that function for performing differential gene expression is also available within the Talk to Data beta system. So you don't have to only perform it on one study, but you can actually perform it on all samples that are specifically related to your filters. So again, this would mean what type of analysis you're performing. So if I'm looking particularly at activated fibros, and I want to compare that from DCM, so dilated forms, and also the hypertrophic forms versus normal quase and fibroblasts in the heart tissue. So that will be my title. These are other parameters that you'll also need to keep in mind. Some of the methods that we normally recommend is t-test. We will be adding other methods as Talk to Data Beta is developed further so that you're able to select what method is more appropriate for the type of data that you're analyzing. And then we'll also sort of include, this is really specific to the question that you're answering. So if you're only interested in your target gene population or your gene set, and you want to be able to see if these genes are sort of upregulated or downregulated at a particular score, you can include your gene set, or you can exclude certain genes that you don't want to sort of have within sort of the heat map results. So again, these are parameters that will change sort of the type of model or the type of experimental sort of model that you're going to perform in Talk to Data. From here, you'll need to sort of add what group you're comparing. So in group 1, what we want to do is compare particularly heart tissue samples. And we want to be able to only sort of select those cells from that tissue. So we'll need to add that filtering for our group 1. And then from here, what I'll add is that I'm particularly looking at cardiomyopathy. So I'll add again my DCM and HCM samples. So I know that group 1 is particularly disease. In the condition group, this is where we'll add again the specific disease filterings that we want to sort of focus on. And the last part will be the type of cell type that we'll be focusing on. So again, this will be the dilated group. And then from there, we'll go to the hypertrophic group as well. From here, we'll go into cell type. And again, this is just, I think, for my filtering or my account, so it shouldn't be an issue. So we'll just add fibroblast cells. Okay. And then our group 2 will be our controlled sort of fibroblasts that are quiescent within the healthy normal tissue. So this will just be our control heart tissue fibro. And then again, we'll need to select the anatomy tissue. Okay. We'll need to select the condition. So this is normal. And then our cell type is specifically fibroblast cells. Okay. So now that we have these cells, what you'll need to also see, it'll show you, I think it's just an issue with my filtering at the moment, but it'll show you how many cells are actually within these particular samples as opposed to the control tissue. So the system will automatically let you know like how many data sets or how many cells we have in each group and what cells are you comparing within your study design. From there, once you add the filtering, you can run the analysis. And once that analysis is run, you can then look at sort of the results that are sort of the status once it's successful. So the first thing we'll want to do is go into our previous results because it will take a few minutes to sort of load that full report. This is where we'll want to then look at what sort of markers are we mainly interested in. So are we looking at upregulation in these activated fibroblasts? If that's the case, we can only filter the entire sort of gene set list to only look at differentially expressed genes that are upregulated within the disease area. So once that is sort of filtered, we can then go to viewing the full report. And this is where you'll have sort of the volcano plot. So this just shows you the extent of how these different differentially expressed genes are upregulated within sort of the heart tissue relating to cardiomyopathy, as opposed to markers that are downregulated in disease. So you'll be able to see these different targets that are very significant. And some ways that you can also filter is by looking at the log fold change. So this is a sort of score that we're able to really define what sort of signature genes are actually at a significant score level differentially expressed within the disease heart tissue. So we can filter those sort of markers to really look at particular genes that I can just see from the list that are very relevant to other studies that were shown previously. So one is the KCNN3. TLL2, we can also see that NRXN3 is quite important as well. And you can go through the list and you can see here that some of the markers that were defined in recent studies, we're also able to see this across other data sets in the system and other samples. So once we select those relevant sort of upregulated markers in sort of the context of fibrosis and activated fibroblast cells, we can then generate a different visualization to really look at how this differs across or versus normal health control heart tissue. So if we apply these sort of selection, this will generate the heat map sort of visualization result, and you'll be able to export this visualization further. So what we can see here is that within the different sort of heart areas, we are able to see that these markers are upregulated specifically in activated fibroblasts as opposed to quiescent fibroblast cells in the normal control subjects. We can also see that POSTN is actually sort of a mediator, so we can see it both within activated and also quiescent fibroblast cells as well. So these are sort of research results that you can also get from this sort of function. And again, this is viewing it by study, but if you want to view it by sample, you can also create a heat map. So you're looking at the different sample variation. So is this result only coming from one study, or can we see it as well across different samples within different data sets? So you can also view that configuration in different ways. So this is sort of what we were trying to answer is how can we sort of investigate these expression profiles of these different differentially significant markers across specifically activated fibroblasts in disease. So that's again the second objective of the session. When we go into the third objective, which is more how can we take this result further? But in this case, how can we really identify pathways that are enriched within this context? So that are driving these activated fibroblasts to lead into fibrosis and also increase collagen and cause other structural changes in the disease sort of form or heart tissue. So for that function, we have this new sort of feature in Talk to Data Beta that was released actually this week, which is the enrichment pathway analysis across all of the studies in the database. So again, this feature was previously only available when you did a one study sort of analysis or a public data set at one study scale, but we can now scale that to all of the filtered samples in the database. So from here, when you're planning to really perform or run an enrichment analysis, there are two features or two selected methods that you'll also need to sort of keep in mind when you're running your study design. So that is whether you're using the set enrichment analysis, so the GCA ranking inputted differential expression score, or if you're using the overrepresentation analysis. So if you're really trying to compare the inputted gene lists across different pathway gene sets and determine if there's a probability or an observation that is very significant for a particular sort of tissue area or a sample. So that's something that you also need to select, and it's specific to the question that you're trying to answer. And then from here in the database, and sorry if it's sort of having some issues, I'll just go back to the analysis results here. And then go back to our Talk to Data Beta. So I'll add our gene. Okay, let's see here. Okay, and then we'll go back to our differential gene expression feature. And then once we have that result, we'll then go into the enrichment analysis. So if we go back to running the analysis, the selected database, this connects you to what type of MCDB databases or biological process pathways you're focusing on, or if it's a KEGG or reactome pathway system that you want to really identify how it's enriched within these different samples in relation to cardiomyopathy. So you can add as many selected database that you really want to sort of run the analysis and you want to explore and how particular markers. So there's also the option where you can select all genes, or you can make it very specific to the targeted signature genes that you were able to identify within these activated fibroblasts that have a very significant upregulation score. So this is something that you'll need to really decide what parameters you're focusing on when you're looking at sort of enrichment analysis from this result. Once you have those results determined, this is where you can analyze the actual result across the databases that we've selected. And this is also determined by different scores. So you can also filter based on the FDR, so the adjusted p-value of these different enriched pathways in the context of these different samples that we selected for the differential gene expression analysis. You'll also be able to see what pathways are actually upregulated or downregulated across, in this case, cardiomyopathy versus healthy quiescent fibroblasts in the normal heart tissue. So to show you a bit how these markers are relevant or these different pathways, I have prepared sort of an easier way that you can see this. So when we go into the different pathways, we can actually see very specific results that we are able to actually gather from past journals as well. So in the dilated and hypertrophic forms of disease, so the specific tissue or fibroblasts within these disease forms, we can see a significant upregulation, particularly in TGF beta receptor signaling, which is a key driver of fibroblast activation, and also how it models or remodels the extracellular matrix. So that actually points into how this increases collagen and causes these structural changes in these forms of disease. Other sort of analysis that we can also gather is that there's also really high enrichment within these activated fibroblasts when it comes to extracellular matrix organization. So again, this also pinpoints to some of the structural changes we were able to see and how this relates to fibrosis or disease progression in this case. Other things that we can also gather is the oxidative stress-induced senescence. So this is another pathway which also contributes to stiffening when it comes to like the structural forms of disease, and also contributes to heart dysfunction. So again, we're able to really pinpoint that these particular markers that are significantly differentially expressed and are upregulated in these activated fibroblasts within disease do show some correlation to these pathways that are also enriched and also lead to these different changes as well. So this sort of activation also relates to inflammatory response. So there's an increase in inflammatory. There's also an increase in cytokine-mediated signaling as well in some of these cells. And this suggests that this can also sort of sustain this fibroblast activation and also sort of the fibrosis, I guess, context of the disease. So these are, again, results that you can perform from the system. And from here, we'll want to sort of, I want to recap what were we able to really identify from this analysis. So some of the things that we were able to identify from these three objectives when you're utilizing talk to data beta is how we can identify these key fibroblast markers in this context. And in this context is within different forms of cardiomyopathy versus healthy control tissue. And we were also able to validate that these particular markers like POSTN, THBC4, and NOX4, which were previously shown from other data sets, we were able to see this enrichment across many other studies in the system. So this is not only dependent on one data set, but we can see this across sample to sample from different studies. The second summary point is how can we identify upregulated genes within a particular group of fibroblast cells? So we were able to run differential gene expression in talk to data beta and really identify particular markers that are related to the alteration or the structural changes in the extracellular matrix, and also other markers that are related to electrical properties in the heart related to these disease forms of cardiomyopathy. So this is actually shown if you generate a larger heat map in the system. This is how we can really gather these particular markers that are actually really specific to these forms of cardiomyopathy as compared to cells coming from fibroblasts that are not activated. So these are quiescent cells. So these are quite important when you're really trying to understand the molecular mechanisms in disease. And the third one was related to how can we perform pathway enrichment on not only one study, but all of these activated fibroblast markers that we identified that are upregulated in disease. So we were able to also correlate this to pathways that really lead to these structural changes and to also increase collagen and fibrosis in cardiomyopathy. So again, these were sort of results that we can gather that I wanted to really showcase. And I think for the next sort of slide, I'll let my colleague Hazel, who really leads the development with the engineering team for Talk to Data Beta, and she'll explain a bit on some of the roadmap and features that we'll have in the next coming quarter.
Oh, yes.
Okay.
Thank you, Alexis, for the excellent presentation. And so, yes, today we use auto data beta to explore the cardiomyopathy. And as you can see that we have some features that we have been using today, which is like gene query, and then like composition analysis, and then DGE, and finally the pathway analysis, as you can see when Alexis show you today. So actually the pathway analysis will be ready and like up and running on your end by the next week. So for now, like we are coming to the final part where we are ready to release this feature. So yes, you will be able to use this feature next week. And then as you can see in the in our roadmap, we also have like several other things that we are doing right now. Like for example, we want to be able to let you to create custom talk to data and also multimodal talk to data, like performing similar analysis but using different type of data, like bulk analytic data or even spatial. And like along the way, we also exploring one thing that we find quite interesting and also important, which is like the cell line analysis. So in here, we also have some like questions to ask you guys about like what other features that you would like to see. And then like, because we are working on the cell line analysis, so would you like to have that features in hand, and what any suggestion that you can give us about the cell line analysis? Please let us know in the survey, and also like other diseases, other case studies that you would like to learn more in the next episode. So please let me know. Okay. And in here, we also received one question from Maria, like how are technical differences between sequencing depths, platforms, and other technical differences between studies accounted for when differential expression and gene set enrichment are done on samples from multiple samples? So basically in here, we use a special unit system we call the ranking system. So based on this ranking system, we kind of like we rank the gene expression, like regardless of the technical differences and also the platform differences. And just to let you know, like in detail about the ranking system, I will share with you like the link to our detailed document on how we transform the data, how we transform and overcome the batch effect in such a big database. Okay, let me just type down here. Okay, so let me down here. Ranking unit. Okay, so as you can see in the chat, I just typed out the ranking unit. And yes, thank you, Maria. And also question from Greg. What database will be used for pathway analysis? So for now, we have Alexis, I think you can show. We have some pathway like the Go, the M6DB, like the I'm sorry, the B6DB, the Go, the CAC, and also Reactome as well. However, in the futures, we will include more and more pathway. And also, Greg, if you have any pathways that you would like to use or any suggestion, please let us know so we can consider incorporate that specific one into our database. So yes, thank you.
So thank you everyone for joining.
Yes, thank you very much, everybody. And please let us know if you have any questions, and also please spend a little bit of time to answer the poll. Okay. So I think if we don't have any other questions, we will end the session here. And yeah, we are looking forward to see you next week. See you later. Oh, for